Okay, I want to introduce the Chemistry 108 students to Dr. Vijay Rangachari, who is a professor of biochemistry in the chemistry biochemistry discipline. And Dr. Rangachari is going to talk to you a little bit about the research program that he has going on in his laboratory. And then we'll ask him a few questions that you as students might have asked him had we been doing this live. So, Dr. Rangachari, I'll turn it over to you.
Thank you, Dr. Masterson. Hey guys, I want to start talking about my research. Before I do that, and I really welcome you to the chemistry and biochemistry. I think a lot of exciting research is going on in the department. And what I will introduce to you is my research. And before I actually jump into this really heavy research topic, I know that you are freshmen. You really want to find out where you want to end up. I kind of give you a pretty, overview of a biochemistry projects that I have in my lab. Many of you, understandably, don't probably won't even recognize what I'm talking about in certain places, but I'll try to tone it down so that you can understand what's going on. Okay, so then the slide, as you can see, I start from a subcellular organelle called ribosome. Some of you may know, some of you, I think most of you have done some biology in high school where you would have been introduced to cells, and ribosome is an organelle within the cell that produces a biomolecule called proteins. And proteins, as many may know, are the workhorses of any function in the cell, right? About 90% of the job and how the job is done in the cell is all conducted by proteins. So proteins are pretty important. So now let's look at how this whole idea of what happens to proteins, so to speak, a birth and death of protein, and what all the things happening in between. So that's what I'm going to show you in this slide. Just I'll walk you through what's happens. Okay. So ribosome, when the protein comes out, and proteins are polymers. And if you may recall what a polymer is, it's like a long string, and the strings chain can be attached to certain beads, right? And if you imagine that analogy, and you can see the proteins are a polymer made of different amino acids, and these amino acids are what we call these beads on the string, right? And when it comes out of the ribosome, if you have a chain with the beads, it can take any shape or form, correct? And that's what you end up getting when it comes out of the ribosome. It is what we call a random coil. It just like a cooked spaghetti, right? It has got different shapes, and there's no particular shape that it can adopt. So it comes out like that. However, the interesting part of this biopolymer protein is that it has to undergo a process called folding. That means it needs to sort of adopt a shape that is specific in many different ways, right? So here I'm showing you the schematic of a protein folding process where the spaghetti-like structure adopts some kind of a cylinder here. So the cylinder is one type of a structure. I won't go into the details, which is called alpha helix. And the arrow marks that I am pointing out here are one type of a very discrete structure called beta sheet. So it's not just always these kind of structure. There can be many different forms that it can adopt. Schematically, what I'm showing you here, it actually undergoes a folding process. And sometimes this folding process is assisted by other protein members called chaperones. Chaperones, as the name suggests, uh, those proteins are the ones which actually help these unfolded protein to undergo some kind of a folding. Okay, so which is very important process in cellular architecture, cellular functions. Now, I want to give you a background of when and what it happened, how the protein chemistry is actually has evolved. In 1950s to 1960s, this is what the biggest achievement in life sciences were. Like if you remember the DNA double helix, great DNA double helix structure, Watson and Crick structure came out in 1953. Around the same time, and proteins were not considered to be a really an interesting molecule at the time, and it was evolving into a field of its own. And that's when this is all happened, the folding process were being found out. Now, why is it important to have a folded protein? It is important because folded proteins are the one which actually carry out many functions in our body. So it is very important that the protein undergoes this folding process to a specific structure that leads to physiological function. Okay, so that's important. Now keep that in mind that every time the folding happens, it is not perfect. The cells also do some mistakes, right? Sometimes it doesn't fold into a structure that is actually going to be functional. It can actually lead to some sort of misfolded protein, which means a wrongly folded structure. Okay. So when it happens, this discovery happened around 1980s, and this misfolded protein can actually then start to stick to one another to form these misfolded aggregates. Aggregates are, you can imagine to be like clumps, right? If you clog a sewer pipe, what happens? Or a blood artery being clogged by a cholesterol, and it leads to mess. And that's the similar kind of analogy you can imagine with misfolded proteins that causes mess in the cells as well. Okay. Now, and these misfolded proteins often are toxic to the cells, and that's those kinds of protein aggregates are called amyloids. That's given a word called amyloids. Amyloids are very toxic material to the cells. Okay, so this discovery happened in 1980s, and that has really opened up many different researchers on the field of protein misfolding diseases. There are about 50 to 60 different pathologies in our body that leads to that is actually based on this protein aggregation phenomena. Now that's one side of the story. Now what happens in the other ones are that if you, well, what I'm not showing you here is that if something goes wrong, like for example in the misfolded protein forms, this can be corrected. The cells have a machinery to correct it, and then put that back into the ribosome to recycle it back to synthesize freshly. And sometimes that machinery also breaks down, and then that's what it leads to a protein aggregation and amyloid formation. But in 1995, very recently, about 25 years ago, um, there's a remarkable discovery occurred in which about half of human proteins are discovered to be remaining in a disordered form. They never undergo folding process, and that's a discovery by itself, which was extremely dramatic and groundbreaking because it's new. Because all till that time, in all biochemistry books, even today, you will always see that the protein has to undergo a folding process for a physiological function. It turns out that it's only half true. Half of our proteins remain disordered form. Those proteins are called intrinsically disordered proteins, which are called IDPs. And there are regions of the protein which are folded, are also which remain unfolded, are called intrinsically disordered regions. Okay, so what do you mean by intrinsic disorder? You can imagine that when you have a particular structure of a protein, they can also have a cooked spaghetti like structure where it wobbles around, it's dynamic, it doesn't adopt a specific shape, right? Now, the question is that it is counterintuitive to what was being proposed 50 years ago, that it has to have a specific structure to have a function. Now, it turns out that these IDPs and IDRs not just functional, but they are multifunctional. Sometimes they do more than one job in the cell. And that is remarkable because the plasticity or the ability for these proteins to adopt different conformations or different structures leads to the idea that it can actually lead to a multifunctional form of proteins. So that's remarkable. That's one discovery that just recently happened. And within that subset of that, I'm pointing out that about 50 to 60% of eukaryotic proteins, that is the mammalian or human proteins, remain disordered. A subset of those IDPs, right, also undergo something fundamentally in well. That is about in 2010, about 10 years old, and it's very recent discovery where these IDPs undergo what we call liquid-liquid phase separation. What does that mean? If you actually use, you must have done or seen vinaigrette, right? Vinaigrette is you take vinegar and olive oil. What happens? They don't mix; they demix, right? So they are immiscible liquids. So in proteins that contain these intrinsically disordered forms, sometimes they actually separate out into droplets within the cytoplasm, right? So it looks like the oil in a water type situation, right? It phase separates to become droplets. They suddenly disappear to become one phase. They come back and they go back and forth. And these kinds of phase separation without any layer separating in the boundary between the other phase is remarkable, because by doing so the proteins can concentrate, they can dilute when it comes out of the two phase, and they can go back to concentrating in one phase. And this is a dynamics that happens only not only most of the time with intrinsically disordered proteins, and that's remarkable. And they form something called membraneless organelles. You may recall in cellular architecture in eukaryotic cells, we know that mitochondria, nucleus, Golgi bodies, all sorts of organelles are separated by plasma membrane, right? I mean, in this case, they can separate out into a membraneless organelles where they can concentrate between multiple components. So that's pretty interesting, and they're reversible. It can come back to one phase, it can go back to that phase. So these are sort of a what do you call this time frame of how the protein chemistry has evolved over time in the last century, right? And you have to keep in mind these IDPs also undergo sometimes misfolded aggregates. Okay. Okay. It can also lead to misfolded aggregates, all of them actually. So there's the dynamics of all of these, how the protein folds into a wrong or right fold, and how the dynamics plays into being toxic or functional is all what my lab is interested in. Okay. So we don't do a lot of different proteins. We do specific proteins which are involved in amyloid diseases. So let me walk you through that. So my lab is a biophysical chemistry. So it's not traditional biochemistry or physical chemistry or even chemistry. So we mix all of these because we work on life sciences. That's why it is biology oriented. But what we do typically is only a sort of a physical chemistry of biomolecules, right? So what do we do? We actually look at the protein misfolding and amyloid formation in neurodegenerative diseases. We work on proteins like amyloid beta, which is involved in Alzheimer's disease. We have alpha synuclein that is involved in Parkinson's disease. We work on a protein called TDP43 that is involved in frontotemporal dementia. You must have heard about a pathology called amyotrophic lateral sclerosis, something similar to multiple sclerosis. And things like these kinds of neurological diseases are quite collective and very closely related clinically. Okay, so that's what we are looking at. We're interested in understanding this protein misfolding and how they aggregate in these diseases. And then, of course, we are fundamentally interested in how these intrinsically disordered proteins work, because it's only things are just starting to pop up about the IDP field. There's plenty more to understand what the mechanics of these proteins are and why they behave the way they do and why they are able to adapt to multiple conformation and how they lead to membraneless organelles and liquid-liquid phase separation and so on and so forth, right? And that is the most important and most interesting form of these. Now, having said this, all of these three projects are interlinked to one another, right? So some of the proteins which actually do for misfolding and amyloid formation are intrinsically disordered proteins, and some of the intrinsically disordered protein do undergo liquid-liquid phase separation, which eventually lead to an amyloid formation as well. So there are dynamics between these that are remarkably interesting for us, at least, right? So if you come to my lab, and I think if you are interested in doing some research, you'll be exposed to these kinds of concepts, and then enjoy doing these fundamental, interesting aspects of physical chemistry of the biomolecules. Here I'm showing you one example of that. So in this picture, this reaction is monitored for about 6 hours, as you can see at the bottom, and it is basically an interaction between two proteins. Both are disordered, by the way. And TDP43 is the one that I told you, which is responsible, which plays a role in frontotemporal dementia. If you look at these here, It looks like a very mundane, simple droplets stuck in a glass plate, right? Which is true, but these are not simple oil and water. These are actual proteins. So you have this combination of these two proteins. Within 6 hours, you can see the small droplets are formed immediately that actually start to stick to one another to form this beautiful liquid droplets. And these liquid droplets have no water, very minimal water in it. So all these things, surfaces are covered with these droplets. On top of this, it's filled with a buffer. So it is a droplet within a water solution, right? So that means these are kind of sticky, hydrophobic or sticky molecules which come together and form this wettable liquid droplets comprised of just these two proteins. It's remarkable how they do it. It's pretty interesting also to look at. So what you will be trained if you come to my lab for your capstone research or whatever you may want to do, whether you want to go to med school or graduate school. If you come to my lab, you will be exposed to what protein chemistry and molecular biology. How do we express the protein in cells? And we use a variety of different spectroscopic techniques that you can actually learn and at least get an idea of what's going on with, such as this fluorescence, circular dichroism, protein NMR, so all of these methods that we routinely use with proteins. And we'll also be talking about my collaborators. We do with mathematical modeling and simulation, and we do cell culture in the lab. And we can also you also get an idea about how to design drugs. These is a long shot in my lab, but we do have a very strong interest in peptide based or non-peptide based drug designs. We want to find out how small molecules can actually interact with these proteins and stop their function or elevate their functionalities, right? So those are the things that you will be exposed to. So I'll stop there, and I think I'm going to I think I've give you a broader picture of what's going on in my lab. If you are interested, Dr. Masterson will have my contact, and you're welcome to contact me. Dr. Masterson, all yours.
All right. Well, thank you, Dr. Rangachari. And I'm sure if you were actually able to be in the class and take some questions, you might hear a few things like this. So this will be your opportunity to kind of respond to potential student questions.
Sure.
If a student was interested in working in your laboratory, at what point would you be willing to take them in? Is there a particular set of coursework that a student has to have before they come work in your lab, or what kind of are your criteria?
Good question. I think the way that I see research for undergraduate is that I don't want any type of research experience to be qualified to come to the lab. This is an opportunity for the undergraduates to come and experience the research the first time. So I'm ready to take even freshmen, but I think you need to go through some kind of safety protocols and safety classes. So I would imagine by sophomore, in fact, I have undergraduate students joined my lab who've done research for 3 years. So I welcome all students, regardless of their background and experience, to come and experience this in the lab for the first time. And I remember when I was doing undergraduation, I did not even know what research was. So nowadays, I think it's a great thing to kids to have this exposure. And there is absolutely no requirement, at least in my lab, that you have to know this to come to my lab. I'm here to train you. Give it a shot. Come here and enjoy research. All I want is the student need to be motivated to learn and identify things, and that's what is important. So as soon as you contact me in a sophomore year, if you are interested, that's great. There's an advantage of joining the lab earlier because within 3 years, spending 3 years, many students do end up getting a scientific publication, which is remarkable wherever you go, whether you go to a medical school or graduate school, doesn't matter, but it's a great thing to have.
Absolutely. Yeah, I agree with that. The other question that I would imagine you would get if you were in the classroom would be, so if a student comes and works in your laboratory, how are they assigned a project? Are they assigned a project all on their own? Are they partnered with a graduate student? Do they have a piece of a bigger project? How does that work in your lab?
Yes, I think so. The way that I do is that when the student comes and joins my lab, almost all the time, whether you join as a senior or a junior or a sophomore, the first semester is always an exposure period. We don't put anybody in any kind of project. So they do assist many graduate students, understand what are the projects going on. And finally, in the second semester, they are oriented towards a project, but not most of the time. Now, I won't say all the time. Most of the time, it's not. It's connected to a graduate student's bigger project. It's a sub project where the student will be closely associated with the graduate student in their work. There are two advantages to it. First of all, I'm not there in the lab all the time to teach them, so the graduate students will be training them partly, and they can ask the questions and see them do things, and they pick up from there. And finally, and the sub project will take shape with the second semester. So what is the direction we are going to take, and the project will be carved. It has also happened that many times the students, depending on the student's ability to process the information, they have been given some really unique small project, standalone project, where the students have done remarkably well, some of the students. But it is not a cup of tea for all of them, right? Sometimes the students want to be assisted. So you are always a part of a sub-project of a graduate student's one. So either way, I think you will have an identifiable project that if you want to write a thesis on for the honors college or not, I mean, you can definitely do that. Yeah.
All right. Well, fantastic. Well, Dr. Rangachari, I want to thank you for taking time to talk with our Chemistry 108 students, and I will definitely put your contact information up at the end of this presentation and encourage the students to reach out to you about potential research opportunities. And so, again, I thank you for your time.
Thank you, Dr. Masterson, for this opportunity. I'm glad to be able to do this to the students.
All right. Thank you. Have a good day.
Alrighty, thanks.